Today, we're diving into a really exciting breakthrough in cancer research. It's a brand new tool that basically acts like a GPS for genetics, and it's giving doctors a much needed guide to navigate one of the most complicated forms of lymphoma out there. I mean, just imagine this for a second. You go through all the tests, the biopsies, the agonizing waiting, only for your doctor to come back and say that your cancer, well, it doesn't really fit into any of the known categories. It's a terrifying and deeply frustrating reality for so many patients, and that is the exact problem that we're going to tackle today. So, the cancer at the heart of this whole puzzle is called diffuse large B-cell lymphoma, or DLBCL for short. Now, the big challenge here is that DLBCL isn't just one single disease. Nope, it's more like a whole collection of different diseases that all happen to look pretty similar under a microscope, but genetically, they're worlds apart. And as you can guess, this makes getting a really precise diagnosis incredibly difficult. To try and make sense of this complexity, doctors have a gold standard tool they use called LymphGen. It's a powerful piece of tech, for sure, but it has one really crucial limitation. See, it was designed to be super cautious. If it's not 100% certain about a tumor's genetic subtype, it just won't make the call. It prioritizes being accurate over being complete, which sounds good, but it means a lot of patients are left without a clear answer. And think about what that actually means in the real world. For roughly 40% of patients, the result from LymphGen is just other. This isn't just a word on a report, it's a massive roadblock. It makes treatment decisions a whole lot harder, and it can actually prevent these patients from getting into clinical trials for new targeted therapies that could potentially save their lives. So, with nearly half of all patients stuck in this diagnostic limbo, you can see why researchers knew they had to find a better way. And that's what led to the development of this really innovative new tool. It's called DLBCL1. So, at its core, what DLBCLone does is it emulates, or clones, the logic of systems we already have, like LymphGen, but it does it with a crucial twist. It's built from the ground up to be more powerful, way more flexible, and to make this kind of cutting-edge genetic analysis accessible for everyday clinical use, not just something for a high-tech research lab. And that's the key, right? This thing was built for the real world. It's panel-aware, which is a fancy way of saying it works with the smaller, more affordable gene panels that clinics are actually using every day. It's also deterministic and reproducible. What that means is a clinic in Vancouver and a lab in London will get the exact same result from the same data every single time. That level of consistency, it's an absolute game changer. Okay, so how does it actually pull this off? Well, the creators came up with this brilliant analogy. Think of it like a GPS for a tumor's genetics. It's all about finding a new patient's exact location on a master map of cancer. Let's just walk through this real quick. First, it creates this huge master map by plotting the genetic data from over 2,000 known tumors. Then, and this part is absolutely critical, it freezes that map, it locks it down. This creates a stable, unchanging reference for everything that follows. So, when a new patient's tumor is sequenced, DLBCL1 projects its data right onto this map to find its exact coordinates. And finally, it just checks the neighborhood. By looking at the known subtypes of its closest neighbors, it can confidently figure out what class the new tumor belongs to. It's simple, elegant, and incredibly powerful. Of course, an idea like this, it all sounds great in theory, right? But the proof is in the pudding. So, did this new GPS actually work better than the old incomplete map? Well, let's take a look at the data. The results were pretty stunning. In one test on over 300 patients, DLBCLone gave a specific genetic subtype to 98 patients who were previously just labeled other. And here's the kicker. It did this without sacrificing any of the accuracy of the gold standard LymphGen. I mean, that's just a huge win-win. Way more answers with the exact same reliability. And this wasn't just a one-off fluke. When they tested it on a completely separate, independent set of data, the results were just as clear. Where LymphGen could only classify about 36% of cases, DLBCLone successfully classified over half of them, 51%. That is not just a small bump, folks. That's a significant leap forward. The team didn't stop there. They also compared it to another modern tool called LymphPlex. And once again, DLBCLone came out way ahead, classifying 74% of tumors compared to LymphPlex's 55%. So no matter how you slice it, this new method is just flat out providing more answers for more patients. So what does all of this mean in the grand scheme of things? Why does providing more answers matter so much? Well, it's all about translating these better numbers into better medicine. You can really see the difference side by side here. We're moving from a system that leaves almost half of patients in that frustrating other category to one that provides way more classifications. We're shifting from needing expensive, comprehensive data to a flexible system that works with the tools clinics already have. And we're replacing these uncertain, kind of maybe results with deterministic, consistent answers you can rely on. And the impact here really is threefold. For patients, it means clarity. It means a clear diagnosis and a much better shot at getting into a trial for a cutting-edge therapy. For doctors, it's a reliable, consistent tool that plugs right into their existing workflow. And for researchers, it's a powerful new platform to push the science forward even faster. Ultimately, what DLBCLN does is act as a vital bridge. It takes these incredibly complex genetic insights that were once kind of stuck in the research lab and makes them practical, accessible, and actually useful in a real-world clinic where they can make a difference for people. And that leaves us with one last, really fascinating question. DLBCL is just one complex disease. Could this whole neighborhood-based cloning approach be the key to unlocking the genetic secrets of many others? It really feels like we've just been handed a brand new, much more detailed map to start exploring.